Hi everyone, my research topic is plastic-free CRF fabrication using natural sources such as arginate. The main cause of agricultural pollution is fertilizers due to its uncontrollable release to the soil. To resolve such issue, controlled-release fertilizers, which is called CRF, were invented to slow down the dosage. However, petroleum-based coating material does not degrade and its remains are critically increasing. Plastic residues in soil caused by input of CRF will be at extreme levels. In addition, most of these substances are non-degradable, thus this would be threatening the soil ecosystem. Hence, in this study, a novel control-release fertilizer was fabricated inspired by biodegradable arginate bead formation used in cuisine. Arginate is a kind of natural anionic polymer. It is extracted from marine brown algae, so it is biocompatible and eatable. Arginate is widely used in molecular cooking techniques such as basics purification. Using this technique, a host eatable water has been developed. The carboxylic group of G block of the arginate forms ionic cross-linking with calcium cation. Usually, the arginate gel beads are prepared by dropping arginate solution into calcium chloride solution. Calcium arginate, as the outer membrane of CRF in our study, was synthesized through ionic cross-linking, varying the gelation time and the concentration of sodium arginate and calcium chloride. The appearance of the bead after drying was observed as follows. The degree of cross-linking was determined by weight, where 7% of sodium arginate and 10% of calcium chloride shows the highest. The sphericity, which can confirm the uniformity of the bead, was measured by optical microscope, which resulted in average of 0.9. Each of these factors was used to form a urea release membrane tuned for versatile growth. The degree of cross-linking increases as the concentration of each solution increases. This is because the higher the concentration of each solution, the higher the probability of forming an egg box, as calcium ions are properly located in the G block of the arginate polymer chain. And the higher the sodium arginate concentration and the lower the calcium chloride concentration, the better the spherical formation. This is because the higher concentration of sodium arginate has the greater the cohesive force. Besides, the lower concentration of calcium chloride, the slower the cross-linking is formed. As the gelation time increases, the characteristics of each sample tend to be saturated. At low concentration of sodium arginate, it was checked that even if the cross-linking time was increases, every encapsulated urea are released within 6 hours. Therefore, the effect of cross-linker was observed after dissolving arginate to the maximum and setting the gelation time 30 minutes. A sigmoid type release trend was observed in each arginate bead, and the release rate was varied by varying the amount of calcium chloride. As a result of increased concentration of cross-linking agent, it was possible to retard the release rate. Accordingly, further experiment will be conducted. Since the maximum solubility of calcium chloride in room temperature is 40%, so we will see if the bead is well formed at very extreme concentration of cross-linking agent. In addition, we would like to see if it can be applied to various crops by slowing the release trend.